So thank you very much. I also want to thank Mark and Andre for inviting me to be here today and provide a perspective, not as an investigator collaborator, but as an NIMH funding perspective on the role of Athina and her work. So I thought I would start out with a few themes. One of them that you heard a lot about today was understanding reward circuitry. So I wanted to start out by just sort of briefly touching on the success that Athina had from a funding perspective. So early in her career, like almost immediately after her entry into research, she really developed a very keen skill in preparing successful grant applications. So she's one of the sort of the model for how we would like to see early career trajectory for investigators in any field. But she was able to obtain both predoctoral and postdoctoral fellowship awards from the National Institute on Drug Abuse, which was really fantastic. And within 1 or 2 years after completing her postdoc, she had her first R01 from NIDA in 2000 called the Neurobiology of Nicotine Reward and Withdrawal, which you've heard a lot about all of the work that she did under that project earlier today. And again, you can see she's had many highly cited papers, many of which were mentioned earlier today. But I just wanted to point out this particular one in which Athina published with George Koob, who spoke earlier today. And it was important because she realized the value of looking at and really establishing the validity, the construct validity and other types of validity and reproducibility and rigor of the tools that were being used to look at and understand reward circuits and processes. And so this was really a key article in which she looked at the way of validating the measures to show that intracranial self-stimulation could be used to establish current thresholds that reflected reward processing, and that she could discriminate these reward effects from the performance effects itself, having to do with the latency measures. So this was really key in the application of this procedure, not only in Athina's work, but throughout all the different types of collaborative studies and other investigators that used it. So again, just building on this theme, so you can see 2 years later now, after she got her first R01 from NIDA, she got her second R01 from the National Institute of Mental Health, which again was really pretty amazing if you think about it, so early in her career. And again, building on the understanding reward circuitry, here she is looking at negative symptoms in schizophrenia and with a focus on animal models. And again, as others have told you, earlier today, the anhedonia or hypohedonia, however you would like to refer to it, she understood and was able to work on the interface of studying treatment development and understanding reward processing, not only in alcohol abuse, but in substance abuse and in psychiatric disorders. So she really was looking at reward processing across the multiple disorders. So if you think of it in terms of what our institute has done and now other institutes are thinking about as well, she was really focusing on domains of dysfunction across all of these disorders, which is really kind of a novel construct. And I just wanted to mention it because, now in 2010, and there was mention earlier today of the research domain criteria, really what Athina was doing was very similar to this approach. And it was before it had a name, but she understood the importance of looking at reward processing across disorders. So in her NIMH grant, she used intracranial self-stimulation really to explore gene expression changes within the circuit. So again, taking a circuit-based approach to understand reward processing. So the next thing I wanted to mention, also has been mentioned many times earlier today, is that really saw Athina as being a catalyst for building translational approaches for treatment development across alcohol abuse, substance use disorders, and psychiatric disorders. And it can't be understated what talent she had in bringing together a disparate group of interdisciplinary researchers that could really address any kind of question that she was really going after. I mean, Nick Cosford talked about it a little bit today, but for example, collaborations between biologists and chemists are difficult. It's traditionally been a very difficult way for the NIH to fund investigators because they speak different languages, they think about things in different way, but she was able to really pull together people and really talk to them and understand the problem, and everybody would bring their expertise and skills to address it. So that was really very important. And so this was mentioned also by several other previous speakers, but I wanted to mention that, so when Bill Corrigal had gone to NIDA to set up a program to really focus on nicotine treatment and prevention, he and I developed a collaborative mechanism called the National Cooperative Drug Discovery Groups, and it focused on both mood disorders and nicotine addiction, and it was an RFA that got put out in 2003. And it's really complicated working on cooperative agreements with the NIH. They're really large projects. They involve NIH staff involvement and overseeing of them, and so it's really different than a research investigated project. And it's really usually complicated and complex, and it requires a real sort of administrative skill to pull everything together. So as John Cryan mentioned earlier, Athina and John put together a team, and part of this was really made a little bit easier because Scripps had already sort of an agreement with Novartis Research Institute in Basel that enabled them, you know, from just a structural perspective in terms of collaborating to move forward with this project easier than if you had just de novo started a collaboration with industry. So that was a really important feature of being able to make this work just from a logistics point of view. But she pulled in a broad group of individuals to address the project, as you've heard earlier, and it focused on GABA B. And the point I wanted to make, and you've seen this already, but I had this in my files from the original funding of the project. This was really a unique collaboration. It was focusing on the GABA B target and was targeting both mood disorders and nicotine. So from the NIH perspective, it's often hard for two different institutes to co-fund a given project because it's just not a straightforward process. But this was a really unique collaboration. It was very much of interest to NIDA, and it was interest to NIMH as well. And so I wanted to point out that there were only 3 projects that were funded in response to this RFA. And so to think that this was one of them, and not only was it one of them, but it involved a collaboration with Novartis, a lot of different investigators, and it involved co-funding from 2 institutes. And it did really well in review, and so that was really a great accomplishment. And I think, you know, Athina had the skill to pull this group together and to work with John, and it pulled in a lot of different people, and it had high-level support from Novartis, like was mentioned earlier, from Paul Herling, from Graham Bilby. It involved Danny Hoyer, Laura Jacobson, many others, as were mentioned. And it even supported postdocs working in Novartis on the project, which again was pretty unusual from an NIH perspective. So I just wanted to mention that, you know, she really was good at doing this, and it was a very successful project. But even so, there were many challenges with the project. And this again is where a very senior, you know, seasoned investigator knows how to deal with it. You know, there came a time where there was a change in the pharmaceutical partners, where they really sort of moved away from this area of research, and they continued to facilitate the project, but we needed to have additional medicinal chemistry support. So Athina went out and found Thomas Finn to take up medicinal chemistry at Scripps to help continue with the project. There were species differences in terms of looking at anxiety constructs and the way the GABA B receptor signaling pathway worked in mice versus rodents, and how that mapped into the pharmacology and the species differences. So there were always little sort of glitches along the road, but they were always solved in a very thoughtful way, you know, with researcher investigators building on this. And so just all of the lessons learned really from working on this project really helped, again, to focus on something that's really important, that you need to have cross-species measures, you know, to evaluate reward processes and look at depression phenotypes in animals and humans. So you heard a little bit from Nick Cosford and also from, Alan Cross about the mGluR23. So there was additional project that Athina worked on having to do with lead optimization of the mGluR23 negative allosteric modulators, and also with the mGluR group 2 antagonists and negative allosteric modulators in depression. So again, you know, she sought out, or Nick says she sought him out, but they formed this really great team that's been working together on the project, again, tapping into domains of anhedonia that are relevant to depression. So, and now again, Diego Pizzagalli touched on this point earlier today, and it was even a bit understated really, but again, so there was another project, it was an RFA developed by NIMH, this time with Lois Winsky being the lead on it, to address translational gaps in treatment development by really trying to identify assays that can be used in both animals and humans, because the predictive assays that were spoken about earlier, you know, they're just missing for certain of our disorders, and so if we want to move forward, we have to find different types of translational tasks that will work. So again, the work that Diego mentioned earlier today, I think that Athina's role in here was really, I think you mentioned a driving force in helping to put together and think about this project, which is really focused on identifying translational brain measures that will allow us to go from preclinical animal data and look at how these measures respond in humans. So the collaboration, again, is a cooperative agreement. It's a different number. You don't need to worry about that. But again, I guess the point is there were only 2 cooperative agreements that were funded in response to this RFA, and Athina and Diego have one of them. And so Diego and his team with Bill Carlison and others at McLean and Andre, Athina, and Aaron at UCSD are part of this project, and they're developing electrophysiological signatures to look at reward tasks, and you see there's many listed there, and they'll be looking at them in animals and then in humans and validating them pharmacologically to see if they respond in the same way. So this is really very innovative, cutting-edge science that we're very excited about, and Athina had a big role in that. So in conclusion, I guess what I wanted to say is that Athina was a very successful scientist from an NIH perspective. She made very impactful scientific discoveries. As you heard, she mentored a number of individuals, and she has a legacy of people that have had the pleasure to work with her. And she's really continued to work at the interface of understanding reward mechanisms across psychiatric disease and substance use disorders, including alcohol abuse. And I wanted to say that the point was made earlier that she had really cared about the students in her lab and her research team and how she was working together to help them move forward without her when she was no longer here. And I wanted to say that that she had an NIMH grant right at the very end. And because both she and Mark and the department chair had spoken about this so much that they were ready to have a new group of investigators move in and take the project forward. So the science that was peer-reviewed and did very well is going to continue with the team that she put together. So thank you very much.